Hi, Aravind. Welcome back to our fourth session discussing the evolution of apoptosis and the history of the discovery of the protein domains involved in the process. In the past three sessions, um, you laid the foundation for our current understanding by giving us an insider view of the chronology of discovery and also the various working hypotheses that existed at each of these stages of discovery. And you also describe how these discoveries added little pieces to the big puzzle, um, which is the understanding of the evolution of apoptosis. One thing that was still a mystery until very recently, and that is the point of discussion for our next couple of sessions, was the biological context of the apoptotic domains in prokaryotes. It wasn't very evident that these were involved in any process parallel to apoptosis, and it was quite difficult to place a finger as to what they were doing. Um, but all that changed after the two Elife papers that we are going to be talking about. The first one of which was published in February of 2020. And it was quite a journey, as when we began, it wasn't like we had nearly solved the puzzle as we felt when we finished the study. So I guess you can take us through this journey and set the background for this.
Yeah, so to start with, sort of to bridge from where we left off, one of the key realizations which came from the studies which we accumulated post the early investigations on apoptosis, which happened in the late 90s and uh, the first 3 years or so of the 2000s, was this principle of biological regulation through opposing forces or positive and negative regulatory controls. And this idea as being a very of negative and positive regulatory forces being integral to a biological system for any kind of decision to be made goes back again to the great JBS Haldane, who, as in those talks which we have heard from the late 1950s, which he gave in India, he expounded after a deep study of physiology and anatomy that the evolution of a system that can set a threshold for some action or to use an information theoretic term to set the filter for noise versus signal can be achieved only by having two opposing forces, a positive regulatory circuit and a negative regulatory circuit. And Haldane demonstrated it in his days by looking at physiological principles in homeostasis or in anatomical principles like how the limb moves and so on. But the same thing translates to biology, that evolution can set these uh, thresholds to discriminate at an allowable level uh, between noise and signal by using two opposing forces, a positive regulator and a negative regulator. And the way this works at the molecular level was something which became clear to us over almost 3 decades of study. And just to give some examples, and then we will bring it back to this context of apoptosis and immunity. Uh, when you have a signal, uh, it's good to have some kind of a road bump or an activation threshold, so that for every little impulse which comes into the system, you don't fire off the downstream process. And one of the things which I realized early through the domains which I studied was that in physiological signaling, especially through cyclic nucleotides, this is a very important key. So you have a generating enzyme like the nucleotide transferase or the synthetase, which generates the cyclic nucleotide. And you have a phosphodiesterase, which degrades it. So this is one way where you have a positive signal coming from the generation of the second messenger and a negative control coming from its degradation. So by adjusting these two opposing forces, evolution can reach the desired threshold at which a signal propagates. Another way to do it is by setting a kind of activation barrier wherein you don't communicate to the generating enzyme, which generates the signal, which may be a nucleotide synthesizing enzyme or a kinase, like say a histidine kinase, by placing a certain filter for or a road bump, which will uh, come in the way of its activation. And this principle became clear when I studied the two component systems and discovered two key domains in them, the HAMP domain and the S helix or the signaling helix, which which always comes between two domains, and it acts as that uh, filter for the impulse coming into one domain flowing into the adjacent domain. And we realized that this that in biology there's a spectrum of these. There are some times when you want to respond very quickly, like say there's DNA damage and you want to respond right away to it. You have something like the SOS mechanism, and there you have a peptidolytic pathway, which is an immediate activation, sensing, and a single peptidase, which activates the downstream SOS response. In this case, the peptidase domain of transcription factors or DNA binding proteins like LEX-A. So another version of this are the one component systems, which are very prevalent in bacteria, where the transcription factor senses the input and directly activates or represses transcription based on the input which it senses. And then you have the two component systems where you have this kind of a roadblock to slow down or to place a filter to judge the intensity of the signal. And in that case, you have a histidine kinase, and the histidine kinase itself is often regulated by these upstream domains like the S helix and the HAM, which set the threshold of the signal it senses. And then it needs to transfer the phosphate from ATP onto a histidine on itself, and that phosphate on the histidine can be dephosphorylated by multiple phosphoesterase activities. And finally, that has to be transferred to the aspartate residue on the receiver domain for the actual signal to propagate. So you have a multi-step process which places an activation barrier, and that can act as a filter. Now, these principles we found in our works in the second decade of the 2000s to extend to the immune systems of prokaryotes, which were subsequently acquired by eukaryotic lineages such as the metazoans. And that came in the form of this nucleotide signaling systems, where you have a, a certain activation barrier in the form that the sensing of the invasive entity doesn't directly trigger the effector, but it first triggers the production of the nucleotide. And so you need a sufficient production of a nucleotide to activate the effector, which happens only when it senses the nucleotide at a certain concentration. And in some of these systems, like some of the CRISPR systems, which depend on a nucleotide dependent activation of the effectors, you have the direct negative regulation coming in the form of phosphoesterases, uh, like the HD phosphoesterase, which is fused to the CRISPR polymerase domain, or the oligonucleotide degrading enzymes, um, which are sort of sensor rolled into the sensor. So you have the CARF domain, which is one of the sensors for these oligonucleotides, also degrading the same cyclic oligonucleotides. And they are sometimes termed the ring nucleases. And there are other ring nucleases too. So that thereby you have a negative regulation of the signal, and thus this interplay between the positive signal in the form of the generation of the oligonucleotide and the negative regulation allows evolution to hit the right threshold which is needed for activation. Now, what we observed was that this kind of a nucleotide based system offers another advantage where, in the event you want to amplify the signal because it has a feed forward character to it, it also provides the option of a tremendous amplificatory capacity, because if you had just a one component system, the signal engages the sensor and then the effector fused to it, which is a distinct domain, usually gets activated. But here, since you have a second messenger intermediate, the activation of one enzymatic sensor of the invasive entity can spawn a large number of nucleotide molecules, which can then amplify the signal. So it simultaneously offers two advantages of setting the threshold and also providing a range of outputs, which can include escalation to a large scale amplification of the signal. So what does all this mean in genomic terms? When we look at the genomes, we found that these systems have some very clear grammar in terms of how they are organized. So you have signal generating enzymes, potential negative regulators in the form of those which degrade the signal, and then effectors. And then you may also have additional force multipliers coupled with them. And these may come in the form of the ubiquitin systems, which are coupled to these nucleotide dependent conflict systems, which are directed against the invasive entity. And one other parallel which we found, which led us in the direction of these current studies, is that you can have a further threshold setting mechanism, which is in the form of a chaperone. And the first chaperone co-chaperone pair which we discovered solved a long-standing mystery. In the late 1990s, I discovered this domain called the Horma domain, which is a common denominator in chromosome segregation, both during mitosis and meiosis. It's found in proteins that associate with the chromosome in both mitotic and meiotic processes. And in a sense, it is a synapomorphy of eukaryotes, which gives you the signal or the evolutionary signal that sex got institutionalized in eukaryotes through the meiotic process. Now, this Horma domain, what it does is to coat these chromosomes by forming oligomers or polymers. And it does so by binding peptides, which may be presented from other Horma proteins. So in a sense, it's a co-chaperone which recognizes the peptide, and it requires an A plus ATPase as its partner, a classical A plus ATPase, which can cause a rearrangement or unfolding of the proteins to unveil the peptide which the Horma domain grabs onto. So the origin of this Horma system had been a long-standing puzzle since the late 1990s when I discovered the domain. And we finally solved it when we were studying these anti-invader immune systems of bacteria, where we found, surprisingly, this Horma A plus ATPase dyad to be located in some of those systems in place of the ubiquitin-like system. So some of them had the ubiquitin-like system as a force multiplier or a parallel mechanism to what we had in to the core nucleotide dependent effector activation system. And others in its place had this HORMA AA plus ATPase. So this was the ortholog of the PCH2 or TRP13 AA plus ATPase that is involved in this process of chromosome segregation, the coating of the chromosomes by the HORMA proteins in eukaryotic cell division and meiotic processes. So what that meant was that there was a further step wherein a threshold could be set by consumption of a nucleotide, that is a nucleotide triphosphate, ATP. So this kind of a platform or an oligomeric assemblage will not happen unless you climb up the slope of energy, and you have to put in energy through the consumption of ATP. So for the final step of effector deployment, you have a further barrier which needs energy to go into it. And this suggested to us that there could be systems which involve a chaperone and potentially its co-chaperone, which could regulate biological conflicts by setting thresholds. So that provided us with a new template to search for in genomic space to discover potential new conflict systems. And that's how the story began. And it led to a very remarkable finding, which I clearly remember the evening when I saw it. And I called you and Max over and just showed it.
Right.
Because it was so striking. And these featured some domains which we knew very well from our more routine protein evolution work, which we had done to apprehend AA plus ATPases. So at that point, we had defined this very distinctive clade of AA plus ATPases, which have some unusual features like those overflow strands, as we call them.
Right.
Which included a class of chaperones such as MOX-R. And in the case of eukaryotes, there's a very conserved protein belonging to this clade of AA plus ATPases, mydosin, which serves as a chaperone in ribosome assembly. And MOX-R, like ATPases, are involved in the assembly of several metabolic complexes in bacteria, as well as unwinding of proteins for degradation in certain bacterial systems, which we had described with Da-Peng during the work on those transglutaminase like peptidases. So the basic principle of these, and then there are the magnesium chelatase systems, which were the classic members of this family, which were well studied. More recently, a former lab member, Balaji, he discovered another such protein, which is a paralog of mydosin, which seems to act as a similar kind of chaperone in the ubiquitination of lipids in eukaryotes. So at the heart of all these is this AA plus ATPase of the MOX-R clade, which acts as an ATP-dependent chaperone, which remodels protein assemblies or unfolds proteins to expose peptides, which may be then grabbed by this co-chaperone, the VWA domain. The VWA domain itself is a rosmanoid, a classic rosmanoid domain. And in the strand 1 position, where you have an active site typically in all rosmanoids, it has conserved aspartate, and then like other rosmanoid domains, you have a crossover, typically strand 4, that is the sheet crosses over to the other side, and at that point, you have the second aspartate, and these together compose what historically had been termed the Midas motif. which is a magnesium binding motif, and it's a magnesium dependent peptide binding domain. So the first VWA domains had been discovered in the very rich von Willebrand factor A protein involved in blood clotting in vertebrates, which was the parent protein from which many a domain has been discovered. But it was VWA which was to have a long history uh, on which we ourselves worked and discovered the first bacterial versions of it many, many years ago in the 90s. So this pair, the chaperone, co-chaperone pair of a MOXR-like ATPase and a VWA chaperone, it's found in many different contexts. And in all those contexts, it's a typical chaperone system which helps in the reorganization of macromolecular complexes through unfolding and of proteins and holding on to peptides in them and allowing the folding to proceed in a certain alternative or preferred pathway. But what was very unusual, so what's typical of the VWA MOXR systems is you just have these two. You may have the A plus in multiple copies, as you see in mydecine and its paralog dynin, which has been repurposed as a motor protein in the eukaryotic cytoskeleton. But it's just the multiplicity of domains of just forms a ring of A plus ATPases. And there's a corresponding ring of these VWA domains, which may be sometimes fused to the MOXR ATPase, but typically they occur as a separate subunit. But they don't have any other decoration. But what was very striking about these proteins, which I observed that evening in course of an analysis, protein analysis, looking for these systems, was that they were fused to a wide array of C-terminal domains. The existence of these longer VWA proteins was sort of known, but nobody had paid any close attention to them or what they even did. So when I looked closer, I found that even between closely related species of actinobacteria, streptomyces species, those domains fused to the C termini of these VWA domains showed a great deal of diversity. And looking at them, it was a peculiar mix of domains. So some of them were recognizable in the sense they were domains which I had encountered before. And most notoriously, they were the APATPases, the famous stand type ATPase from the apoptotic system. So those were very common. And they may have a further run of TPR repeats. And in some cases, you had just TPR repeats directly fused to the C terminus of these VWA domains. But there were also a wide range of other domains beyond these AA plus domains, which were fused to this VWA domain. And that the composition of those C-terminal domains, which were fused to this VWA domain, was extraordinarily diverse. A very wide complement of domains could be seen, which we illustrate in that paper of ours. And that network is truly staggering. It's just the domains fused to this VWA, and the complexity of the network is striking. So within the first hour or so of me poking around with it, I realized there was a fairly striking diversity. And that's when I called you and Max. And you could see for yourself the striking nature of the diversity. And each of you all could see some domains which you had worked on previously.
Right.
Sort of your favorite domains.
Right.
They were all fused to the C terminus of this VWA domain. And just to name a few, apart from the APA TPAs, it included the FPSK domain, which we had earlier studied in chromosome segregation systems across life and viruses too. There were protein kinases, there were other cell division related proteins like MIND, and then there were these nucleotide related signaling enzymes, such as adenylyl and guanylyl cyclases, more generally the CNMP cyclases. There were the REases, the restriction endonuclease, like endonuclease domains, and a mysterious set of glycosyl transferases whose true significance we only understood later, and of course caspases, again hearkening back to the system of the apoptosis systems, the slog domains, and then domains which might process uh, or process NAD plus, the nicotinamide dinucleotide, like the DR hide and macro domains. So I'm naming just very few of these here. There were many more, and there were several domains which sort of stood out there, which mystified us. Uh, so what struck us at that point was that there was such a uh, diversity of these in terms of cellular functions uh, in which they may operate. So it's not like you found one type of cellular system, nor were they immediately reminiscent of the effectors which we had seen in the other conflict systems. And as we discussed in the last episode, that there was this principle of effector sharing between disparate biological conflict systems, which we had uncovered. But some of those effectors we were shared with this system too, but there were many others which we never saw as effectors in that system. But what reminded you of those other biological conflict systems was this architectural diversity. So the MOXR and the VWA domain were constant components. They were always there uh, in these systems which we uncovered, but it was the C-terminal portion, C-terminal to the VWA domain, which was extraordinarily diverse. And its diversification, even between closely related species, reminded one of the polymorphic toxins, where you have the toxin tip continuously varying, as we discussed the last time. So right then, it told us that we were looking at something which was under positive selection for diversity. And it is a hallmark of biological conflict. Only when you have an arms race between closely related species, you see these domains diversifying so uh, rapidly. And what's more is that these domains in this diversified repertoire of domains, C-terminal to the VWA, they did not fit in any one functional category. So whatever explanation we had to come up with for this could not pass through a very simple explanation of that they are effectors which are attacking self nucleic acids or invasive nucleic acids or self proteins or something like that. Now, the next, then we decided that this was a remarkable system and it deserved all our attention, and we put our the full for all the members of the lab who could engage were brought in. Gurmeet was asked to systematically study it, and we started digging into this. So at that point, it became clear that these came at their core as three gene operons. So two of those components were genes coding for the MOXR, which always stood by itself, but it had some interesting features which distinguished it from the other MOXR proteins in the form of of an N-terminal extension. And the same applied to the VWA, which had a distinctive N-terminal extension. So we could isolate these systems from all regular MOXR VWA systems through that signature. And the VWAs in this system were, of course, further defined by a C-terminal extension, a distinct domain which came after the VWA domain. And beyond that, which we call the VWA linker domain, or L. And then beyond that lay this astonishing array of effectors or diverse domains. At that point, we still weren't clear that they were effectors, but that was the idea building in our mind. Because they parallel the effectors and the diversification of effectors in other systems, biological complex systems, which we had studied. So those lay further to the C-terminus. And it was not like there was just one of those variable domains fused to this VWA protein. There was a very wide range of them. Some of them may have one, some of them may have several of them tandemly fused, one after the other. And there was not even a necessarily obvious logic to these, to the things which were fused to it. That is, you may have a kinase, you may have a GTPase, you may have caspasers. So it was all kinds of stuff, FPSK, RNA binding domains. So we were pretty astonished by that diversity when we encountered it. Then the third component was something which we had never encountered before. In fact, before I go to the third component, I must say that there were up to 11 domains which could be fused in that variable region. And in
one polypeptide.
In one polypeptide, yeah. Right. And the mean was about 3.5. So with a modal peak at around 4, and you could go up to 11 distinct domains. So that's just to give the reader an idea of how these are. And there was the third component, which we had never seen in any other VWA MOXR systems before. And that third component was, again, a feature which set these systems apart from all other VWA MOXR systems. And we termed that the VWA MOXR associated protein, or VMAP. Now, this VMAP had a constant C-terminal domain, which we termed the VMAP-C domain accordingly. It had a very variable middle domain, which we called the VMAP-M. And then to the N-terminus of this VMAP-M was again a variable region, but those were globular domains. VMAP-M tended to be almost always alpha helical and small. But the N-terminal domains were globular, and they came in a few broad flavors. So the most common version of those N-terminal domains were peptidases. So you either had caspases or trypsin, like peptidases, as the most common version. And more rarer versions were papain-like peptidases or the transglutaminase-like clade. The next most typical type was something which generated a nucleotide signal. So you had CNMP cyclases, GGDF domains, and others in that same vein, which may generate a signal from NAD, such as TIR domains and the related DR hides, and the polynucleotide, or sorry, the purine or pyrimidine nucleotide phosphorylases, the PNPase domains. That's a family of domains, which in this case, we could predict to have a specific chemistry, that is clipping off the base and releasing a signal in the form of a clipped base, or perhaps generating a derivative from NAD, like cyclic ADP ribose or ADP ribose. So you had peptidases, you had these enzymes which could generate soluble signals, or you had a set of domains which were to then lead us to something very important, but in the first step, we didn't fully understand them, called the EADs. We simply call them EAD for effector associated domain. And we uncovered over 10 of such domains. So there was some diversity in them. So this was the basic morphology of that VMAP, or the third component. You have these enzymatic domains, either peptidases or soluble second messenger generating enzymes, uh, or these EADs, which we'll return to later. A central, typically alpha helical, highly variable small domain, the VMAP M, and finally the C terminal uh, constant domain, the VMAP C. So this operonic organization allowed us to quickly piece together how this complex may be constituted. That there was, in all likelihood, uh, the typical VWA MOXR chaperone co-chaperone complex, which are forming toroidal rings at the heart of this. And then that is joined by this third component in the form of the VMAP protein. And then there are the signaling enzymes, which are associated with the N terminus of the VMAP protein. And there are all these highly variable domains associated with the C terminus of the VWA uh, protein. So that's the basic anatomy. So we could imagine that there is a ternary complex being formed, because there are three distinct components at the genetic level: VWA, MOXR, and VMAP. So this gave us the template of how a system which we term the ternary system could exist. And in a sense, it gave us a new grammar. So like in physiological signaling, you have one component signaling systems or transcription factors, two component systems, and now we had a ternary system. And as a working hypothesis, two things caught our attention. One was that, or maybe three things caught our attention. One was that when we built phylogenetic trees of the VMAP, VWA, and MOXR components individually, we found that they showed a very congruent phylogeny, which meant that each of these components, three components of the ternary system, went together. You didn't have a mixing and matching of VWAs from one system going with the MOXR from another system. So there was a vertical evolution in the sense of the core conserved components: VMAP-C, the MOXR, and the VWA. What we could infer from that point was that they formed this tight complex, which supported our basic working hypothesis. The second notable point which we observed was that we did an entropy analysis using the Shannon entropy derived from the alignment, and measuring the mean entropy as well as the distribution of the entropy. And that showed that these components were evolving at very different rates. So first, the MOXR component was not fast evolving. So what that meant was that the MOXR component was not doing something very different. The selection on it was to just to stay the same. Whereas the VMAP and the VWA component, so in fact, we used as a positive control, say, the DNA polymerase 3 beta subunit. And the entropy of that and the MOXR from the same organisms was the mean entropy was roughly the same. Whereas the VWA and the VMAP-C components were much more fast evolving, significantly more fast evolving. And what that implied was that there was some kind of diversifying selection on them. So they were at the interface of something which was driving them to diversify. And this is, again, a strong signal for a system being involved in a biological conflict, because it parallels what we have observed with other effector systems, like, say, the polymorphic toxin systems or the CR effectors, which we spoke of two episodes back, uh, where you have constant regions which are typically low entropy, and the these parts which are actually engaging in the conflict, which are more fast evolving. So it suggested that the VWA and the VMAP C components were closer to that threshold of, or they were closer to that interface of being actually engaged in the conflict.
Mhm.
Uh, so this was the second notable point, which strengthened our contention that we are looking at some novel systems involved in biological conflicts. And the third and the most dramatic thing which caught our attention was that uh, these showed a very peculiar phyletic distribution. So it was not like any bacteria had them, but a systematic survey showed that they were predominantly present in bacteria which had something which could be termed real multicellularity.
Right.
Now, in the first two episodes, we had discussed about how, by definition, an organism can be seen as a superposition of unicellular and multicellular states, but we can distinguish something called more real multicellularity when you have differentiation. So, not all cells are doing the same thing. They are not only aggregating together, but there is a division of labor between the cells. So it may even be that some of them are sacrificing their fitness for their kin, which are actually doing all the reproduction. So we see this only in some bacteria. The most prominent of these, where we first discovered these systems, are the actinobacteria, which are multicellular, which are often multicellular. Streptomyces exhibiting some of the most complex morphology uh, among the bacteria, and cyanobacteria, which are again can be morphologically very complex. And as both of us know very well, we have spent some time picking those colonies, which are macroscopic. So you can consider a cyanobacterium like Anabena as a macroscopic multicellular bacterium
Right.
with those nice globular colonies which look like those green marbles.
Right.
And then we also found them in other lineages which where this multicellularity is less common, but there are a few representatives which might be so, forming complex filamentous structures or rosettes. For example, Bacteroidetes, some of which most Bacteroidetes would be seen as typically unicellular, like those which inhabit the human gut. But then there are some which form these complex filamentous structures with a differentiation. And the same can be said of planktomycetes, which form these rosette like structures,
Right.
and chloroflexi, which form the filaments again, differentiated filaments. And the one place where you don't see multicellularity that often are the proteobacteria taken as a whole. But then there are some lineages of them which show some of the most complex multicellular forms, like delta proteobacteria.
Right. Right.
And in the same delta proteobacterial lineage, you can see the whole spectrum from something like Delovibrio uh,
Right.
or uh, uh, this Aneromyxobacter, which are not really very complex. Delovibrio is even parasitic. But then you have the Myxobacteria proper, like Myxococcus or Stigmatella, which are some of the most structurally complex bacteria in terms of their multicellular uh, aggregation. So, and likewise in actinobacteria, you can see that spectrum. You have things like Micrococcus luteus, uh, with which we have spent some time in our youth. Uh,
Yeah, especially as a
uh,
contaminant.
yeah, as a yeah, well, since it inhabits our palms,
Right.
they are full of Micrococcus luteus. Even if you think you've washed them. So whatever you touch, if you touch a plate, you can get Micrococcus luteus in it. But Micrococcus luteus is not particularly multicellular. And
Right.
then you have something sort of midway, like Corynebacterium or some of the mycobacteria.
Right.
And then finally you have Streptomyces or Streptiverticillum, which are really multicellular in by any definition of the term. So uh, what we observed was that within a lineage, like say delta proteobacteria or actinobacteria, these non-multicellular ones did not have these ternary systems,
Right.
whereas those which were multicellular were enriched, sometimes having multiple copies uh, of the say of these ternary systems. And when there were multiple copies, those C-terminal variable domains fused to the VWA domain tended to vary greatly. So it was not like the same copies, the same version was repeated.
Right. Right.
So
Yeah.
yeah,
Oh,
so
the one small aside that actually I remember when we made this statement and when we wrote this paper up, one of the reviewers asked us for a formal statistic, and it
yeah,
was it took us some time to actually go through every bacterium that was there in our list. Some we knew we had some
yeah,
data, but some we just had to go through tons of papers to see if there is any evidence for multicellularity. And it's a strange thing that we do not have in this day and age with such great imaging facilities. We still do not have a proper formal database of multicellular prokaryotes. And
right. I think there was a decent paper. I don't know if it was on the Coulter lab or
right,
in PLOS, which had a good discussion on multicellular bacteria, but there was no that there's no systematic database, and essentially we ended up creating one for this
yeah,
purpose.
yeah, and only
And
then we could apply the formal statistic that you had done.
exactly. And we did the formal statistic. In this case, I think I would say that this uh, that particular ask by the referee was a good thing,
Right,
because even when we submitted it, I told you that someone could come up with that objection.
right, right.
Uh, and sure enough, that referee came up with it, and I think it in the end, what the labor which we went through, splitting it sort of three-way between you, me, and Max,
Right.
it was a time well spent that we could build up this good database for formal statistical testing of significant enrichment of these ternary systems in multicellular bacteria. And the net result was we added one figure to the paper, but I'm pretty happy with that figure that we got to show those multicellular bacteria with the statistics.
Right, 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 right. Yeah, yeah, yeah. Sorry, just as an aside.
Yeah, no, I think that was an important point to stress, since that will keep coming up. So here we had to sort of uh, take a to summarize where we stand. We have these systems which we term ternary systems because they have three conserved components, with two of them having these variable domains fused to a conserved core, which show this very distinctive phyletic spread with they are almost signposts. If you find a ternary system, you can almost say that organism is multicellular. And one thing which came up was we found them in a small number of archaea, both euryarchaea and members of the TAC uh, group, that is Crenarchaea. They were the ones which we found were in Crenarchaea, but they belong to the broader clade of the TAC clade of archaea. So a priori, we were not aware of what these archaea, what it meant to find these in archaea. We were quite conversant with multicellular bacteria from our prior bacteriological explorations, purely from the viewpoint of natural history. But these archaea were unfamiliar to us. And then when we were doing the systematic collection of the multicellularity status, when we looked into them, we found that the concealed in the obscure descriptions of these archaeal lineages was the statement that they form these complex laminae or spheroidal aggregates, suggesting that these are a certain type of multicellular archaea. Of course, one of them was previously well known to us as being multicellular, that is methanosarcina, which forms those multicellular aggregates. So this was a very good positive, or it was a sort of a jackknife, you can say, where we didn't know that these were multicellular a priori, and later when we did the investigation, we found that the presence of these systems correlated with this multicellularity in the archaea. So anyhow, so what we had were these systems with a dramatic variability between even closely related organisms in certain domains, which were found in archaea and bacteria, which had a which were significantly enriched in multicellular archaea and bacteria. So what did all this mean? And the clue to this came from a very careful analysis of those C-terminal variable domains, uh, which we found in fused to the VWA component. And this is something which we are adept at. It's not necessarily something people would immediately understand, because it's not just detection of the domain. That's something which must be stressed. So we found many different domains which are found in various other cellular systems. So they are not necessarily found in conflict systems. For a very good example to just harp on the point is FTSK. So FTSK is part of the cell division apparatus, where it acts as a pump which helps pumping the chromosomes into the daughter cells in bacteria. So why would FTSK be fused here? Another such was tubulin, the tubulin FTSZ-like protein. So tubulin, again, part of the cytoskeleton. When so these are two cytoskeletal proteins. Just to give you the contrast, on the other end, we had things related, which were very regular conflict endonucleases, like REases, the NA1 restriction enzyme was another, and several nucleotide binding domains and RNA binding domains, such as the S1 like OB fold domain, S1 cold shock domain like OB fold domain, KH domains, and some superfamily 1 helicase domain. But then when we looked more closely, we found that many of the enzymatic domains like FTSK or caspasers or protein kinases, the MIND like GTPase clade NTPases, while being enzymatic in this particular context, they were inactive. That is, there was a disruption of the catalytic sites. The catalytic residues were substituted, suggesting that they were inactive. So that was very odd, to say the least. And the second factor, which was very unusual, was that you may have coupling of things which are functionally unrelated. For example, the FTSK may be linked to a CNMP cyclase in the same polypeptide. And that's not, and again, an inactive CNMP cyclase at that. And that's not a kind of association which we encounter in the typical cellular systems where FTSK or CNMP cyclases come. So this meant that there was something very strange going on. But before I sort of unravel this, I think what I should do is to proceed with the other systems which we uncovered. So now that we had this grammar of the ternary system, we went back to the genome and looked for others which may fit such grammar. And what we found was that there were at least 4 other systems or 5 other systems now, which could be called these MOXR-like ternary system, MOXR VWA ternary system. So they had a MOXR and a VWA component as constant elements. But the third component, unlike VMAP, was different in each of these new ternary systems. So one of them, interestingly, instead of VMAP, had something which we call iSTAND for inactive stand. And uh, it was a stand type NTPase, which we have discussed quite extensively in the previous episodes. But the ATP binding sites, both Walker A and B, were disrupted. So it was an inactive stand domain. And in this case, those variable domains, which look like the variable effectors of conflict systems, tended to be fused to this iSTAND component. Or alternatively, they were fused to that linker domain, which comes C-terminal to the VWA domain. And that occurred as a separate gene, which was adjacent to the core ternary set. And one more of these systems had, again, a very interesting set of interesting combination. It had the MOXR and VWA as usual, and the VWA was fused instead here to C-terminal beta propellers. And the third component in place of iSTAND or VMAP was FPSH. So these were FPSH like AA plus ATPases. Now, those who are connoisseurs of AA plus ATPases like we are would know that FPSH is a very distinct clade of AA plus ATPases. They are more of what you may call the classical AA plus ATPases, closer to the proteasomal AA plus ATPase. So that took the place of the third component. So you had the FPSH like ternary VWA MOXR systems. And there were at least 2 more which we found. And in the case of this FPSH like systems, also coupled to them were one of 3 alternative systems. One of them involved some novel effectors, which were very variable proteins, or at least what we thought were effectors. We'll build that case more as we go along. Another had a potential biosynthetic module, which we predict to synthesize a modified peptide with a radical SAM enzyme, which uses adomet or S-adenosyl methionine to modify peptides. And such peptide modification systems have been widely observed in bacteria, but here it's tightly coupled to this system. And then the third potential effector system coupled to this FPSH type MOXR VWA system was a complete triligase ubiquitin conjugation system of the prokaryotic type. So finally, we had the ubiquitin system, which we have seen in eukaryotic conflicts, now again tie up to a prokaryotic conflict system. It sort of brought back some things which we discussed in the previous episode, our study on the UB prokaryotic UV systems. So those came and docked in with these systems. So if this sounds complex for you all, that we are just beginning our story here. Be warned. There's much more to it. And as I said, there are at least two more of these MOXAR VWA systems, but I'm not going to discuss them in too much detail. At least one of them is pretty much like the second of these systems. That's the iSTAND systems. And so we simply called it the iSTAND 2 systems. The third of them, the fifth. The fifth one is this one with iSTAND 2. And the sixth of these had something which we called a TERNS, or a ternary system associated protein, as the third component. So the third component can vary between systems. The MOXAR and VWA are constant across all MOXAR VWA systems. However, the third component, VMAP, iSTAND, iSTAND2, FPSH, TERNS, and a beta propeller, that is yet another of these. So all these are the third component which can vary. And then within each system, there is a variability of a variable domain collection, which we are calling effectors, though the case has only been partially built for why they are effectors. And so this is as far as these VWA MOXAR systems go. But these gave us a template that there can be this kind of ternary regulatory mechanism where you have three components participating in the core of the regulatory system. So that, using that grammar, we went back to look at genomic neighborhoods, wherein you don't have to have a VWA or a MOXAR, but something else takes the place of these three components. And through that mechanism, we discovered a whole series of other potential ternary or ternary-like systems. And one of these, which sort of hews closest to this general syntax, which we saw in the VWA MOXAR systems, were what we call the DO-GTPase system. So here the DO is for double GTPase. So they always, or they tend to have two GTPases. That's why double GTPase. And this is a very distinctive GTPase, which is found only in these systems. And these GTPases are in turn related to the septins, the paraseptins, AIG, GMAPs, dynamins. So that's a whole clade of GTPases, which I had described previously. And many of them are involved in immunity and regulation of apoptosis in eukaryotes, in metazoans and plants. For example, the GMAP systems are important regulators of immunity in metazoans. AIG, TOC, they are seen in plants, AIG involved in plant immunity. And then you have the interferon-induced GTP binding protein, the IIGPs, which are related to the dynamins, which are also members of this clade. So very interestingly, we now had the first prokaryotic versions of those which might be involved in an immunity-related context. So you have these two DO-GTPases. And they had, in addition, two more components in the list. One of these components we termed GAP1 for GTPase associated protein 1. And there were two flavors of it, GAP1N1 and GAP1N2, which differed in the in sequence in terms of their N-terminal domains. And that distinguished two types of this DO-GTPase system. The third component in the GAP1N2 systems was a fibronectin type 3 domain. That was the what you could term the constant third component to complete the ternary system. So you have the GTPase, the GAP1 protein, and the fibronectin type 3 domain. And this fibronectin type 3 domain containing proteins, they often were fused to variable uh, components, again pointing to that effector variability architecture. And in the same operon, which were fairly complex operons, you had other components, some of which were constant and others highly variable. Among the more constant ones were components like tubulin, uh, HSP70, GRPE, and DNA-J homologs, suggesting a whole HSP70 like chaperone system was incorporated. And protein kinases, in this case active, and proteins with secreted VWA or cell surface VWA domains, very different from the MOXAR VWA pair. Here, these are VWA domains which act like receptors or sensors. So this already is giving you a taste of the complexity of these systems. These are the gap 1 and 2 systems. And the gap1 N1 systems similarly have the DOGTPase as one component. They have gap1 as the second component, and the third component, which we term GASH, and that takes the place of that fibronectin type 3 containing protein in these systems. And they are coupled in the operon with highly variable components, which have a certain constant part and the variable part, which are typical of effectors. In this case, the effectors are those which we have found in other conflict systems. So that gave us a clue that we are indeed looking at biological conflict. So this is for a ternary system wherein the grammar is similar, but the components themselves are replaced by something else. And another notable point here is that these DOGTPases show the hallmarks of being close to the membrane. Apart from the fact that the septin, dynamin-like clade of GTPases, GMAPs, most of them work as oligomeric complexes in the inner leaf of the cell membrane. These DOGTPases often come with TM spanning domains, which suggests that they are being anchored to the membrane. Or other proteins in these systems, they are fused to TM spanning domains. So we are looking at some conflict scenario, likely close to the membrane. And to move on, as I said, there's enormous amount of diversity here, which is not for the faint heart. You have to have considerable eye for detail to understand these systems. So I will be very brief with the other two systems which we found as being part of this general ternary-like mechanism. And what we found as the basic grammar here is something which I'll first expound before I move on to this. So the way we interpreted this was that there are different kinds of variable components. So like the VMAPs, as I mentioned, they have this middle domain, VMAP M, which is quite variable. Now, if you have the I stands can be quite variable too, in terms of when you measure it using entropy. So that suggests that there is some pressure on them to diversify, whereas the MOXAR remains relatively low entropy. And what this suggested to us is that this third component is integral to sensing a moving target, an evolutionarily moving target. And that what are common moving targets inside cells? They're invasive entities like viruses. So in all likelihood, this third component acts as the sensor for something which is invasive. And the stand NTPases, as we discussed in the previous episodes, bind they have evolved from proteins which bind DNA as part of the replication system of both cells and mobile elements. So the stand could potentially be while it's inactive and hence not carrying out its ATPase role. The very fact that you have an inactive stand could mean that it could still serve as a sensor for invasive nucleic acids. And we know from eukaryotic systems that one of the ways that viruses are sensed is by sensing invasive nucleic acids, like invasive RNA through the helicard uh, helicase in metazoans, which is a helicase fused to card domains. Uh, so these I-stand domains could act in a similar way as being sensors for invasive nucleic acids. It's also possible that some of the other ternary uh, components, the non-I-stand third component, could function as direct sensors of proteins. So what we have in our model here is that this unique component which distinguishes these MOXR VWA systems from all the uh, garden variety chaperones doing cellular housekeeping functions, so to say, is this third component, which is likely sensing the invasive entity. And the sensing of that invasive entity triggers a threshold setting mechanism, which is mediated by this MOXR VWA path. So only if the signal is intense enough, you have an activation through the consumption of ATP. And that causes an unfolding, which in all likelihood involves the unfurling of these effectors, which are then held in a configuration which is ready for activation by the VWA domain, which binds peptides. And it doesn't end there, because as we discussed, like say in the VMAP, VWA MOXR type ternary systems, you have a further signal coming from the end terminus of the VMAP. And this could be a peptidase, or it could be a nucleotide signal, cyclic nucleotide or nucleotide NAD derived signal like ADP ribose or cyclic ADP ribose. So it's not just that the MOXR VWA is enough in most of these cases, but it just is the first step in the activation. And then you need the second step in the form of proteolysis or the nucleotide derived signal, which then deploys these effectors. In the case of proteolysis, we imagine that either the trypsin-like peptidase or the combination of the caspase-like peptidase and the alpha-beta hydrolase, which always comes with these caspases in this system, cleaves off those effectors fused to the C-terminus of the VWA component, and thus allows them to diffuse and do their effector business. Now, in some of the other ternary systems, it could act slightly differently. One possible mechanism could be that they activate a UB system, like in the FPSH type MOXR VWA ternary systems, or they activate the synthesis of some kind of a toxic peptide or a signal peptide. Signal in the sense it acts as a signaling peptide. Now, there have been studies in actinobacteria which shows that the production of antibiotic metabolites is accompanied by apoptosis. So you could imagine a triggering of cell death to release these peptides. And whether the peptides have a direct antiviral role or whether they act as a signal that then induces a certain immune response to the invader remains to be seen. But one thing which became clear is that these peptide signals may be even found in the VMAP-like systems because they are often coupled to a further operon it's distinct from this ternary core, but it occurs as an adjacent operon which is coupled to it, with often with its own transcription factor, which is CNMP binding transcription factor related to CRP. And that additional module is a module which contains a biosynthetic system for a peptide which is being modified by this radical SAM enzyme. So it has a metallopeptidase which is cleaving off from the precursor protein, a C-terminal region, which we could locate as being the peptide which is likely to be modified. And then that is modified by the associated radical SAM component. So these are reminiscent of some previously described peptides which have been seen before. For example, those which are termed the FXXX systems.
Right.
And so these, like say, one such radical SAM associated with this is spasm.
Right.
So that so these are that kind of a peptide where the radical SAM likely catalyzes linkages between lysine, tryptophan, or cysteine, and thus it makes a cyclic derivative of these peptides. And there is these have been catalyzed. These peptides, these peptide cyclases have been characterized to a degree in previously in the literature. But what exactly they are doing is unclear. And in this case, there's good evidence that they are participating in this immune mechanism. So that's for one type of action. But how do these effectors elicit their action? That's the next question, which before I move on to other such systems, that's one thing to cover. As I said, the so-called effector position often features enzymes which are found in other effector systems like REases or the PIN endonuclease, which we saw in the antitoxin system. So those are more likely to act as direct attacking effectors, in the sense they attack the invasive nucleic acid, or they attack self-RNA to prevent the replication of the invasive entity. But then there are all these unusual domains in the effector position, like the inactive FPSK, the inactive kinases, inactive MIND. So what we propose here is that viral proteins, they, in order to carry out their functions, and it's well known from a lot of studies in bacteriophages from the earliest days, that they recruit cellular proteins as subunits. For example, some bacteriophages utilize thioredoxin as a subunit of one of their enzymes. They may modify cellular proteins by ADP ribosylation, or transferring acetyl or other acyl groups through the action of GNATs or GCN5 like N-acetyl transferases, or cleaving them through peptidases. So cellular proteins from various cellular systems, which are often called host factors, are recruited by viruses to do their business. Now, you can imagine that if you have a version of a protein from these cellular systems, which is inactive, it can serve as a decoy for the cellular protein which the virus uses. So we propose that several of these effectors, which are inactive variants of proteins in regular cellular processes, act as these decoys or dominant negatives, which interfere with the host factors which the virus uses for its replication or transcription or any other thing it needs to do. So that's why you have this very unusual set of effectors, the inactive enzyme effectors. But then we found a third class of effectors which solved a long-standing mystery in cyanobacteria. These are the so-called FGS domains. The name FGS comes from a very unusual thing, formyl glycine modifying enzyme. It has nothing to do with that. It is a peptide binding domain having a C-type lectin-like fold. And these were first discovered in a class of diversity generating systems from bacteriophages, wherein the tail proteins of these bacteriophages undergo diversification uh, through an adjacent reverse transcriptase, a mutagenic reverse transcriptase, which uses a short template which corresponds to part of this FGS domain and replicates it and reinserts it into the genome, thereby diversifying it. So these systems discovered by Miller very long back, they were initially found to diversify these phage tails and thus enable them to cover a wider host range or evade host immunity developing from mutations of surface proteins which prevent the tails from sticking to the host surface. And Miller's lab had found many chromosomal versions across bacteria and described them as undergoing variability in the same fashion through these associated reverse transcriptases as the cellular version, as the phage versions. But nobody really knew what they were doing, especially in cyanobacteria where they're abundant. But I think we finally solved that problem because we found that they were the most common C-terminal effector fused to the VWA component in these ternary systems. And suddenly it hit us that now we had the bacterial analog of the metazoan adaptive immunity. So just to recap, in metazoa jawed vertebrates, they have an adaptive immunity which proceeds through hypervariability, which can happen through VDJ recombination mediated through a domesticated transposon. Or you can have uh, direct mutagenesis mediated by the AID or AID-related AID-ApoBec like deaminases. In the case of the jawless fishes or jawless vertebrates like the lamprey, you see the same thing being mediated by this deaminase and the gene conversion like process. Uh, but here, instead of acting on the immunoglobulins, it acts on the VLRs. So what we propose here is that this third type of effector is essentially an analog of this hypermutating adaptive immunity in cyanobacteria and some other bacteria like planktomycetes, which also have this FGS domain, wherein the FGS domain is mutated by the action of the reverse transcriptase, which often occurs as the adjacent gene, the mutagenic reverse transcriptase. And that diversification now allows it to recognize new invaders. So by if the invader evolves away, there can be mutagenesis and it can thereby adapt to a new type of invader. So it's not the same, but it is analogous. That's why it's not homologous, but there is the analogy here is in that hypermutagenic process happening within the cell to recognize invasive entities. So what we see here is that these ternary systems combine the principle of high regulation through setting of these roadblocks in the form of thresholds or activation barriers, which need to be overcome by the utilization of ATP. But that alone is not enough. you need a further step, which may involve proteolysis or a biosynthetic process, like synthesis of a peptide or a mutagenic effect or a hyper mutated effector, which then recognizes the invasive entity. But what happens thereafter? And here is where the clue came from some of these highly variable effectors in the VMAP like ternary systems, where I mentioned the presence of APA TPS and somewhat more less common are the narked ATPases. So finally, we had a very direct link between a potential immunological process and this process of apoptosis. So here we imagine that some of these effectors, like say the tubulins found in the DOGTPS system or the HSP 70 like systems, again associated with DOGTPases, the same tubulins occurring in the VMAP ternary systems, and the superstructure forming repeats like beta propellers and TPRs, they could all form these multimeric assemblies, aggregates, which could kill the cell or physically provide a barrier for the spread of the invasive entity. In many ways, reminiscent of these prion like templated assemblies of members of the death like superfamily, like pyrin um, or card in the metazoan systems. So, this kind of uh, aggregational direction to cell death was seen here. We could predict that directly from this analysis. And that suggested that finally we had the evidence that at least in these multicellular prokaryotes, the same components like APA, TPases, and caspases were part of an immune mechanism which was sensing invasive entities and responding to it by generating these assemblages or by direct deployment of effectors, which could trigger apoptosis to prevent the spread of the invasive entity, the virus, through a multicellular assembly. And why might you have these only in multicellular organisms as opposed to unicellular ones? Because by definition, they having their kin next to them uh, in a very institutionalized way uh, as part of their uh, very mode of existence, they expose themselves to invasion of other cells once one cell gets infected. So here that old principle of kin selection, which we described, can kick in, and if one cell sacrifices itself, it can prevent the spread of the infection through the rest of the colony or the rest of the multicellular assemblage, and thus limit the infection. So the apoptotic road to immunity was pretty important here. So to round up this part of the story or this chapter, I'll just touch upon two things. So using this general template, we found at least two other systems, as I mentioned, beyond the VMAP, or sorry, the VWA, MOXAR systems and the DOGTPA systems. We actually found three other systems because one was reported in a different paper with Max, which are the CATRA systems, which are,
Right.
the CATRA comes from the caspase. So here we have this EACC1 system, which has a constant component in the form of a membrane-bound sensor protein, which we predict to be a sensor protein, likely a membrane-bound invader. A constant component in the form of a caspase, that's the second constant component. And then the caspase is fused to an extraordinary diversity of other domains, which cover a similar diversity as we saw in the VMAP MOXAR VWA systems. And those are again predicted to be effectors based on this diversity between closely related organisms. And so here we see a peptidolytic caspase cascade, which again reminds us of the proteolytic cascades activating eukaryotic apoptosis, unleashing these effectors. In a similar vein, the other system which we found involved a trypsin and a nuc-A as the core components. And this was again likely a proteolytic activation happening with the trypsin releasing nuc-A, which is a HNH fold endonuclease, which we found as an effector earlier in the polymorphic toxin systems, which we discussed before. And so here again, perhaps a targeting of self-RNA is the core mechanism, but there are other effectors which are recruited to this system, which come through a different mechanism. And that will be the final part of this edition of the story or this episode. So what's that final component? If you had paid close attention to this very complex picture which I have been painting, I mentioned that the VMAP, in addition to enzymatic domains, such as cyclic nucleotide or nucleotide derivative generating enzymes or the peptidase enzymes, had these mysterious domains which we called EADs or effector associated domains. So these EADs, they show a very characteristic genomic organization. If they come fused to the N terminus of a VMAP, almost always you have adjacent genes which have a second EAD containing protein, which a code for a second EAD containing protein. And that EAD is fused to further effector domains. And these are again very typical effector domains, which may include Atpases or caspases and a slew of other effectors which we have found in other conflict systems. So what you see here is that there is an EAD-EAD coupling. If there is an EAD fused to the VMAP, you have an EAD encoded by a gene adjacent to it. Or you have many other EAD encoding genes elsewhere in the genome of this organism. And there's one actinomycete which held the record, which I think had about 21 different EAD genes elsewhere and adjacent to the core ternary systems. So these EAD genes, their products are very characteristic. They have this constant domain, which is the EAD, and there may be up to one of 12 currently, 12 or 13 EAD domains. And they are fused to either N or C terminal variable domains, which are characteristic effectors. Now we found the same EAD kind of association, not just in the VMAP and other ternary systems of the VWA MOXAR type, but also those of the DOGTPase type and this nuc-A trypsin type. So in each of these cases, the same types of overlapping set of EADs reappeared with a similar configuration. There's an EAD fused to the core constant components, and then there's an EAD fused to variable effector domains. And in a phylogenetic analysis, typically the EADs which are adjacent to each other in the genome or from the same organism tend to group together. So it's a lineage specific development of these EADs, a lineage specific expansion or a lineage specific duplication. And that is a telltale signal that these are forming homotypic assemblies. So there is a homotypic interaction between these EADs. Now, the most surprising thing which struck us was that at least two of these EADs happened to, in profile searches, happen to be related to the death-like superfamily of metazoan apoptotic domains. So after 2 decades, 2 long decades, we had finally found the prokaryotic homologs of uh, the notorious death-like superfamily. And they were found in these ternary systems as these coupling domains, which we had through the argument which I just presented. Uh, the signals, or they had the features of being homotypic interaction domains which are recruiting effectors. So we had done this all prior to finding this relationship. And then we find this relationship of two of these EADs, and we name them BDLD1 for bacterial death-like domain 1 and BDLD2 to highlight their relationship to the death-like domains from eukaryotes. And they were no different in their architectural features from these other EADs, uh, which we had uncovered in these systems. So what it suggested was that this principle of homotypic interactions bringing effectors, which we see in the metazoan apoptosis system involving the death-like domains, could now be extended to these EAD domains found in these bacterial systems. Thus, we completed much of this analogizing between uh, prokaryotic immunity systems uh, of this ternary variety and the eukaryotic immunity systems which involve apoptosis. And the key point was that the majority of these ternary type systems which we described had a statistically significant occurrence in multicellular uh, prokaryotes, primarily bacteria. So that meant that the apoptosis process was indeed a hallmark of this multicellular state of existence. And it showed that indeed all these uh, effectors and uh, regulatory components like APA, TPases, which play a role in eukaryotic apoptosis, come together in a similar fashion in these multicellular bacteria to mediate apoptotic mechanisms as part of highly regulated uh, immune mechanisms, which come under this broad rubric of what we call the ternary systems. So it's very remarkable that the same vocabulary has been used for immune and apoptosis related processes, both in eukarya and in the two prokaryotic superkingdoms. So it shows that this vocabulary has a very strong functional signal to it. And there is a very strong grammar associated with it. So as soon as you can assemble that grammar, these domains can fall in place to enact processes like apoptosis in conjunction with immune mechanisms. So I think that sort of more or less rounds up this fairly complicated story of the ternary systems and how we were able to place the homologs of prokaryotic homologs of eukaryotic apoptosis systems finally in a mechanism that we predict as acting in a multicellular context to do comparable things in prokaryotes.
Yes, yes, but I mean, it was still not the end in the sense that I think in the second part, we achieve a higher degree, we achieved a higher degree of clarity on many nuances of these systems. And in fact, I think a full evolutionary picture of how they came, how they were sort of perhaps acquired, or how did they come to the eukaryotes, also became clear in the second study. And also the fact that there are many more such systems, even in eukaryotes, that we were totally unaware of, and which are quite exciting in terms of them being adaptive and you know similarly configured.
Right. So this is the first part
Right.
of this story, and given its extraordinary complexity, uh, I think we will stop here, and we'll
Right.
have a further episode where we will put the capstone, which will
Right.
have, I must warn any listener who has lasted this far, uh, an equal amount of complexity,
Right.
but it'll sort of put the nail in the block with respect to this whole issue of the origin of these apoptosis systems in these bacterial immunity contexts.
Right. Right. So I think let us just stop here, and
Yeah,
we'll continue in our next session. So let me
yeah.
just